get contaminated because of environment, because of machines, because of materials, because of cross-contamination, because of other APIs or other products, and because of handling procedures. And for this purpose, to avoid traces of the other products or other materials in next product, we have to establish very good cleaning procedures. And these procedures have to be validated procedures. And cleaning validation concept is covering how to validate the procedures and what shall be the parameters for detecting or freezing the limits for the routine activities. So why cleaning validation is so important? Particular attention should be accorded to the validation of cleaning procedures. This is according to WHO. Then according to PICS, cleaning validation should be performed in order to confirm the effectiveness of a cleaning procedure. According to FDA, means US FDA, the data should support a conclusion that residues have been reduced to an acceptable level. US FDA is not saying that you have to remove entire traces or it is possible or not possible. This cannot be guaranteed. So they are saying acceptable level we have to maintain for the traces in other products, or you can say traces in next procedure. Possible contaminants of the product residues from last product, cleaning agent residues and breakdown. So it may be cleaning agents which you are using, it may be T-Pol or detergent or anything. So traces of the same may retain in the machine, or it can be stick to the machine parts and it can carry on in next product. Airborne matters, lubricants, ancillary materials. So lubricants which are being used in machines, specifically in compression machine or some machines where there are chances of going these lubricants in product. And that is the reason we are forcing for food grade lubricants for compression machines. Decomposition residues, decomposition of any kind of product. If product is not stable enough and it can decompose during the process because of heat or moisture, and that residues, decomposition residues can be stick to the machine or stick to the equipment. So bacteria, molds, or pyrogens in, and these are the microbial threat or microbial contamination, possible microbial contamination. Validation strategies on cleaning validation. Product contact surfaces we have to consider. After product changeovers, we have to consider cleaning validation. Between batches in campaigns, we have to consider cleaning validations. Bracketing products for cleaning validation, periodic re-evaluation and validation. Now, bracketing products means grouping the products for cleaning validation, means similar category products, similar kind of molecules, similar procedures with similar molecules. So this kind of things we can bracket for cleaning validation. So cleaning validation protocol should include objective of the validation, responsibility for performing and approving validation studies, description of the equipment to be used, means contact parts and non-contact parts to be categorized during in protocol itself. It should include interval between end of the production and cleaning and commencement of the cleaning procedure. Cleaning procedure to be used. This is first point is also important because interval between two productions and cleaning. So it shall be defined that how much time you are going to keep that equipment ideal. Otherwise, in many cases, there might be chances of microbial contamination, like if you are producing liquid products and you are keeping this for more time, so it may generate or it may be there might be chances of microbial contamination. So cleaning procedure time intervals must be considered, and the time taken for cleaning procedure is also most important. So cleaning procedures to be used. So what kind of cleaning procedures you are going to use, manual cleaning procedure or CIP cleaning procedures, or you have to define which cleaning agents you are going to use for this cleaning of the product. Any routine monitoring equipments used. So you have to define which equipments you are going to use for cleaning purpose. Number of cleaning cycles performed consecutively. This shall be also defined in protocol. Sampling procedures used and rationales for the same shall be defined. Rationale for the all procedures, in fact, we have to define many times what we are doing. We are considering and assuming something based on our past experience, and we are putting this in protocol. And when auditors are asking for what is rationale behind setting up this procedure, we don't have answer. For example, when I'm asking you peoples that which cleaning agent you are going to use, you peoples are just saying, no, we are going to use only water. But what is rationale for the same? If you studied anything for your molecules, if any molecule you are having which is not water soluble, then what you are going to do? If you are using some waxy material in your ointment or external preparation area or liquid area, if you are using you are manufacturing certain elixirs which can be oil part can be stick to the uh, oily syrups, if you are manufacturing like vitamin syrups and all, vitamin A syrup and all, and if it is sticking to the machine, what rationale you are having by making the statement that you are going to use water, only water for the cleaning the purpose? So that cannot be rational and you cannot justify the same. So solubility of the API must be considered while using cleaning agents, and all APIs cannot be soluble in water. So ultimately, you cannot claim that I'm going to use only water for cleaning the equipments or cleaning the beans or cleaning other utensils and parts of the equipments where product is in contact. So sampling procedure, cleaning procedure as well as sampling procedures shall be used, and that shall be rational procedures. You cannot go with a thumb rule that we will, based on our experience, we will use this one and industry is using this one. So I am using this one. I was using this in my past companies, so I am using this one. That cannot be rational. Rationale must be defined based on your products which you are going to take, and based on that only your cleaning method and cleaning procedure must be evaluated. And that is reason most of the companies are using TPOL or any other solution which you can say neutral solution. So this neutral solution helps to remove the traces of the product, which it may be water soluble or it may not be water soluble. Sampling locations must be clearly defined from where you are going to sample for cleaning validation. It cannot be only here, this is surface where my hand is reaching and from that only I'm going to draw the sample. It shall be defined based on the critical parameters like difficult to clean area we have to consider first, and that difficult to clean area shall be identified as a rationale for sampling location. Record of cleaning validation should include data on recovery studies, analytical methods, including limit of detection and limit of quantitation, acceptance criteria and rationales for the same. When revalidation will be required, must have management and QA involvement. Management commitment and QA involvement is necessary for cleaning validation activities. Now, analytical methods, including limit of detection and limit of quantitation, means you have to validate your analytical method, which is being used to test the residue of the cleaning or used to test the sample which is drawn during cleaning validation of the machine. Results and reports, cleaning records signed by operator, checked by production and reviewed by QA. Final validation report, including conclusion, shall be there. Now we will come to the part how and what will be the procedure for cleaning validation studies. So personal manual cleaning methods are difficult to validate. Cannot validate people. These are most important part. Manual cleaning methods are very difficult to validate because person to person it may change. Water quality every day may change. He may apply force on certain areas. He may apply pressure on certain areas for cleaning. He may casually clean the any areas of the equipment. So manual cleaning methods validation is very difficult. Cannot validate peoples, can measure proficiency of the peoples. Many times we are saying that our peoples are validated. So you cannot validate any person because you can only measure proficiency based on your experience. Say you are having 5 operators, from 5 operators, operator number 1, 2, 3, 4, 5. First operator is having very good proficiency and their analytical results during his cleaning part or cleaning samples are very good, and other operator number 5 is not cleaning that much efficiently. So proficiency and training you can impart to the person, must have good training to the persons, so that these persons will be trained efficiently to act on their operations, and they can clean the equipments very effectively, so that equipment can be cleaned without any traces. Must have effective supervision. So it's not like that every time we have to believe on the operators. Our operators are 10 years old, and 10 years, from 10 years, 20 years, they are working with our factory, and we are believing in that. Not like that, because effective supervision must be there, so that they shall not change the procedure of the cleaning by their own. They shall not change the cleaning agent. They shall not change the cleaning method. They shall not change the sequence of the cleaning. Like suppose we have defined any sequence for any equipment, how to open the equipment, how to dismantle the equipment while cleaning. And during dismantles, he shall not use any reverse process, or some part he has to open first and he is opening that last. So effective supervision is also most important part during cleaning validation. So microbial aspects of the cleaning validation, it shall be included in validation strategies, analysis, risk contamination, analyze risk of contamination during cleaning, also before cleaning and post cleaning also. Consider equipment storage time. Equipment should be stored dry. Sterilization and pyrogen contamination shall be considered. Now this equipment storage time is most important part in cleaning validation, because many times, can you see the screen?
Yes.
Yeah. So many times we are just standard way we are saying and defining that equipment storage time shall be 72 hours, but it cannot be. You have to do full-time study for every equipment, and whole-time study shall be done before commencement of production in your area. Means when that equipment is ideal, that time also you have to consider whole time, that how many days the if equipment I am keeping ideal, so within how many days there are certain contamination, dust or some other particles, or say some microbial contamination is happening in the equipment. Then during the empty runs, during the load runs of the equipment, during qualification also you can define the equipment whole time study. And during production, again, I'm saying during the process validation part, you have to consider equipment storage time, that after completion of the production, if I am keeping that equipment in dirty conditions, in how many days it will create microbial contamination, or the product which is sticking to the equipment is degrading in how many days. Plus, after cleaning, when I'm storing the equipment, so how many days I can keep the equipment. As such, you have to define in your SOP of the cleaning or protocol of the cleaning validation, that if that equipment is ideal after in clean condition for say 7 days, 8 days, 10 days, based on the whole time study. If you are not finding anything during 8 days, 10 days, you can keep that time also. But when you are taking this equipment again for use, that time, whether it shall be cleaned again or not, that shall be justified based on the rationale. And you cannot just take this that before you clean the equipment before use. This is very vague statement. So we have to define certain time frame, and after that time frame, we have to clean the equipment before use. Sterilization, in case of your manufacturing sterile products or degradable products, you have to, which are having more susceptible to the microbial growth, that equipment shall be sterilized and depyrogenating during the before use, and it will avoid contamination, pyrogen contamination in the product. How to sample? We can sample swab or fast sample, rinse fluid sample, placebo samples. The sample transport and storage conditions should be defined, because this is very critical sample which you are drawing, and this sample and extraction of the sample from the swab shall be defined. Rinse fluid, the containers for the rinse fluid, closing of the container, then covering of the container and transportation of the container from production to QC department shall be defined very carefully. It shall not get contaminated in between, and the transportation of the sample shall be very carefully done. Swab sample, direct sampling method, reproducibility, extraction efficiency shall be defined for swab samples. Document swab location. Document the swab locations from where you have taken the swab for testing. Disadvantages, inability to access some areas. That's why I'm saying you have to define the hard to clean part of the machine, and from there you have to try to take the sample so that that representative sample will give you realistic result of the cleaning. Easy to clean part can be easily cleaned, and from that part, if you are taking samples, that will definitely come positive and that will give good results. Assumes uniformity of contamination surfaces. Uniformity of contamination surfaces. Must extrapolate sample area to whole surface. Rinse sample indirect method. This is indirect method of cleaning sample collection. Combine with swabs. You can combine this with swab also, useful for cleaning agents, residues. pH, conductivity, and TOC will be most effective ways to analyze the rinse sample. Insufficient evidences of cleaning. Sample very large surface areas. Basically, rinse sample we are taking after cleaning. Means cleaning has been completed. All the cycles of the cleaning has been completed. Say 3 wash, 2 wash you are giving to your equipment. And after that, we are taking this rinse sample. And this sample will be collected, and this is covering very large surface areas. And that is one advantage. And testing of the same can be done through pH, conductivity, and TOC, because only we have to check in rinse sample that cleaning agent residues have to be studied. And if you are using some detergent or some TPOL-like solutions, then these cleaning agents may be showing some pH, conductivity, or TOC changes, TOC reading changes in the sample. Analytical methods shall be validated for cleaning validation method, cleaning validation sample testing. Must be sensitive assay procedures, HPLC, GC, or HPTLC. You can use pH, conductivity, UV, or in case of microbial, ELISA microbial test, you can conduct for cleaning validation sample testing. So analytical method shall be validated with precision, linearity, selectivity, limit of detection, or LOQ, limit of quantitation. Then recovery by spiking, consistency of the recovery. Setting limits, regulatory authorities do not set limits for specific products. Now, many people are saying that there are certain regulatory guidelines, and according to guidelines, these are the limits. So there are no limits specified by any regulatory authority for any product for cleaning validation or cleaning samples testing. So logically based, it has to be decided based on the molecule, based on the method of analysis. We have to define our limits logically. Limits must be practical, achievable, and verifiable. Unnecessarily, we shall not give very stringent limits, considering that coming to this limit and it will come to that limit. So don't assume like this. Set your limits very practical, achievable, and very verifiable. Based on the calculations, you can set these limits and follow that limit. Allergenic and potent substances shall be considered first for cleaning validations. Limit setting approach shall be defined very carefully based on the product, based on the molecule, based on the characteristics of the product. Uniform distribution of contaminants not guaranteed. While setting the limits, we have to keep in our mind the product which is going to contaminate, or the material or the particles which are going to contaminate your equipment or next product, that is not assured that they will distribute very uniformly. It may be at a corner of equipment, or it may be stick to certain surfaces, but cannot guarantee that uniformly this will be distributed uniformly. And that's the reason our cleaning method must be very, very efficient. So decomposition products to be checked. The products which are getting decomposed because of heat, because of certain moisture or environmental condition must be considered and checked before setting the limits. Setting limits, cleaning criteria, 3 criteria you can say: visually clean, 10 ppm criteria in another products, 0.1% of therapeutic dose. So these are basically standard criteria based on the industrial practices and history of the industry. These criteria are generally being considered, and this is having rationale, that this is having history, but this is not written in guideline that you have to consider this much criteria. You may consider these criteria for cleaning as a cleaning criteria. Visually clean is the first criteria which we shall follow very carefully. Can be very sensitive but needs verification every time. Used between same product, batches of same formulations. Similar products are there and similar molecules are there. In that case, or similar product but different batches, in that case you can use this criteria for visually cleaning, because if these products are carrying some particles or some material in next product or next batch, so that will not going to affect any therapeutic value of the product, or it is not considered as contamination. So we have to clean the equipments, but visual cleaning procedure can be implemented for this purpose. Illuminate surfaces, spiking studies we can use. It's like illuminate surfaces means you can use some battery like things to check the surfaces whether it is showing any particulate contamination or not. Setting limits 10 ppm, it is having historical background. In some poisons regulations, it has been mentioned that 10 ppm, not more than 10 ppm shall be carried in for the products. Pharmacopoeia's limit test you can consider for the same. Assumes residue to be harmful as a heavy metals. Useful for materials for which no available toxicological data, not for pharmacologically potent materials. So this can be useful for the materials for which there is no toxicological data or toxicological study have been done for the materials. And this cannot be used for very potent materials which are pharmacologically potent, like hormones, then like anti-cancer drugs or any other steroids or microsteroids, you can say. For these materials, you cannot use this criteria. Setting limits not more than 0.1%. Very simple method you can use for clinical validation, that is proportion of minimum daily dose of current product criteria over into maximum daily dose of subsequent product. Need to identify worst case products, and you can set this that not more than 0.1% shall be carry over in other product. This can be a very easiest criteria when you are not having any PDA data with you, or when you are not having any specific data for your product with you. So other issues, clean in place CIP systems, you are using these systems must be validated very carefully. But this is giving very good results than your manual cleaning, because once you are having CIP system and you are validating the same with pressure, with sprinklers, and with with the movements of the sprinkler and the pressure of the water and all these its parameters, if you are validating once, then there is no need of validation, there are no chances of variations in the cleaning method. So then manual process, this is very good process. Placebo studies, detergent residues, composition should be known for detergent residue if you are using detergents. Scrubbing by hands, this is the issue may create during scrubbing by hand. Questions for the GMP inspectors to ask: how is equipment cleaned? Are different cleaning processes required? How many times is a cleaning process repeated before acceptable results are obtained? What is the most appropriate solvent or detergent to be used? At what point does the system become clean? What does visually clean means? So this has to be defined in your SOP or during the cleaning validation, because this may be asked by the GMP inspector or where the auditors are there. So these questions have to be keep in mind. The manufacturer needs a cleaning validation strategy. Assess each situation on its merit. Equipment rationales must be developed. Equipment selection, contamination distribution, significance of the contaminants, visually clean may be all that is required. Now for cleaning validation, you may establish certain criteria based on the concept of PD or ADE. So permitted daily exposure limit PD represent a substance specific dose that is unlikely to cause an adverse effect in an individual is exposed at or below this dose every day for a lifetime. Acceptable daily exposure means represent a dose that is unlikely to cause an adverse effect of an individual is exposed by any route at or below this dose. Every PD and AD are effectively synonymous. So PD and AD, these are synonymous and you can use this, but understand this has to be done if your product is new and you are doing clinical and toxicology for this product. Then only you can establish these factors, because below you can see F1, F2, F3, F4, and F5. These are various variables which you have to consider for establishing PD and ADE limit. Now, for if you are having 500 products with you, you cannot do this study for every product. It is very costly affair and it is very difficult to get the things. And nobody can establish this value based on theoretical results. So what you have to do, you have to purchase whatever PD and AD values published in the literature by some other company or by some R&D, by some scientist or by some regulatory authorities. This is again a costly affair. So instead of doing this PD, AD level permitted, when you are going for regulatory after 2 years or 3 years, that time you can go ahead with PD and AD value. Right now, you can go for this 10%, 0.1% and visual inspection basis. So if you are going for this PD studies, then it is very costly. And again, I don't think so for 500 products, if you are going for and purchasing the data, then that is okay. And then you can have values, ready-made values in your hand, and you can, based on that, you can establish your cleaning validation method. Another thing I will say, I will suggest that don't go for immediately for very high profile study for every product, because whatever products are already established in market that you can, and you are not going for regulatory, don't go for this study of PD or ADA for the same. Macro, yes, you can do this macros, but for this also you need PD studies. Then minimum batch sizes you will require, safety factor of the product you have to consider, and then standard therapeutic daily dose of the product you have to consider. Again, you need for this PDA value of the product, and based on this you can calculate MACO, maximum allowable carryover of every product. Whatever products you are going to file for semi-regulatory and regulatory, that you have to must go for this product. Last aspect is the risk-based approach. So during cleaning validation, health-based exposure limits, good knowledge management, that is ICH Q10, you can read for the same, and risk-based approach, that is ICH Q9, risk assessment for the operations, cross-contamination strategies. So during risk assessment of your process, of your equipment, of your product, you can consider cleaning validation factors also, that what will be the possible contaminants and what will be the possible risk of contamination in your product. And based on that, you can define your cleaning validation, cleaning method and cleaning validation. Bracketing for cleaning validation group typically based on equipment range, cleaning procedures, dosage forms, rationale explained in SOPs or cleaning validation documents, groupings from which worst case will be selected. Any product that does not conform to the bracket must be validated individually. Means while doing cleaning validation, don't go for product wise, individual product wise. Go for campaign based, that these are the 10 products which are having similar category, similar dosage forms, similar equipments being used, similar cleaning method is being used, similar procedure or that process is being used. So based on that, you group the products in category wise, and this bracketing will give you results, and don't go for individual product screening validation. Worst case determination shall be done for the product based on the potency and the batch size of the product, contact area, product solubility study, product toxicity release mechanism shall be studied while considering worst case determination. So again, product solubility is most important part in this. So worst case process conditions, campaign length, number of batches or time elapsed, dirty hold time study shall be done, minimum limits for manual cleaning, time for cleaning steps, temperature, CIP and CIP programs you have to validate. So these are all about cleaning validation. So you can ask now your questions on this so that we can conclude the topic.
सर वो पीडी वैल्यू वाला निकालो ना स्लाइड.
Just a second.
हाँ, उसके आगे. हाँ, ये जो पीडी वैल्यू हमको निकालने का है वो तो मतलब डेटाबेस ही है ना.
Yes, this you see, if you want to establish PD value for your product, you have to give your product for toxicological study. You have to get the acceptable or permitted daily exposure limit as per as toxicology is considered. Is what will be the maximum daily exposure limit for your product that has to be studied through toxicology study only, and it has to be go for clinical evaluation. Otherwise, if your product has been studied by some laboratory, or if data is available with some other clients or some other laboratory, and if they are going to give you with so taking some money, then that data you can use. But in that case also, you have to have similarity of your product, dosage form, and the product characteristics.
और इसके पीछे वाली स्लाइड है उसमें मैक्रो का कैलकुलेशन है, उसमें भी पीडी आती है, तो उसके लिए
you have to establish it.
लेकिन ये F1, F2, F3, F4, F5 है, वो तो अलग-अलग स्टडी है ना, रेट माइस.
Yes, based on that only PD value will be defined, and that you cannot do in house.
ये हम इन हाउस मतलब ये डेटा कलेक्शन बेस पे ही कर सकते हैं ना.
How you can do this? Who will give you authenticated data? You cannot do this based on the internet studies.
और ये जो NOAEL का जो वो कैसे निकालते हैं?
It is novel concept is for new drugs, and that's why I'm saying this PD/AD cannot be done in house, and you cannot only take data from internet and based on that you will do this. Novel study you have to do in clinical laboratories only, and they have to first consider the maximum toxicological dose. In novel study, what they are doing, they are considering maximum toxicological dose, the dose on which adverse effect is coming, daily permittable dose, daily maximum highest dose, minimum dose. That's all studies will are being done during clinical trials and during BE studies also, and based on toxicology studies, and based on that, this novel value and F1, F2, F3, F4, F5, these factors are being decided. You cannot just search that what is F1 factor for paracetamol. This may be different for different formulations or for different processes.
सर एक्चुअली हमको यहाँ क्लीनिंग वैलिडेशन स्टडी करवाने की है हॉर्मोन्स प्रोडक्ट्स के लिए, तो क्लीनिंग वैलिडेशन स्टडी
purchase this data or send your product to laboratory. They will give you PD value, AD value, and micro value.
सर अभी तो क्या है, यू का करना नहीं है, तो कुछ सजेशन दो ना कि फॉर ROW अप टू
मैंने
6
बोला ना
कंट्री।
आपको, आपने पहले स्लाइड्स में दिखाया ना वो कि आपको यू गो फॉर दिस क्राइटेरिया।
नहीं सर, बट इतना मैको तो ढूंढना ही पड़ेगा ना, क्योंकि
तो
वर्स्ट
ये
केस भी तो
आप
आइडेंटिफाई
लेबोरेटरी के पास से ऑथेंटिकेटेड परचेस करो ना, सिर्फ ढूंढने।
सर, बट मैं क्या बोल रही हूँ, पीडी के अलावा भी तो मैको ढूंढ सकते हैं ना।
वही मैंने बोला मैडम कि आपको जो उस प्रोडक्ट का जो मैक्सिमम डेली डोज है, मैक्सिमम अलाउेबल कैरी ओवर लिमिट है, वो एस्टेब्लिश करने के लिए उसका एडवर्स रिएक्शन का डोज कैलकुलेट करना पड़ेगा ना। तो वो डोज
वो मुझे
कैलकुलेट
मिल
करने
जा रहा
के
है।
लिए, हाँ, वो करने
राइट।
के लिए आपको लेबोरेटरी के पास से उसका स्टडी तो लेना पड़ेगा ना। विदाउट स्टडी यू कैन नॉट गो।
मुझे ये बताइए सर, WHO और TRS क्या कहता है?
ये सभी
वो
WHO का ही गाइडलाइन, अभी जो प्रेजेंटेशन मैंने दिया ना आपको,
बट वो
इस
मैको का बोल रहा है या वो ये सब क्या?
नहीं, वो WHO कुछ नहीं बोल रहा है। दुनिया का कोई भी गाइडलाइन आपको ये सजेस्ट नहीं करता है कि आपने पीडी से करना है, मैको से करना है, मैक्सिमम अलाउेबल कैरी ओवर से करना है,
नहीं सर,
या
नहीं
वर्स्ट
सर,
केस करना है।
EU स्पेसिफिकली बोल रहा है कि पीडी रिपोर्ट चाहिए, और EU स्पेसिफिकली ये भी
ठीक है,
बोल
मैंने
रहा है कि
आपको
आपको
क्या बोला कि
टॉक्सिकोलॉजिस्ट का पीडी
if
चाहिए।
you are going for regulated, then you have to do
हाँ,
all the studies. There is no excuses.
अग्रीड, मना नहीं कर रही हूँ मैं सर, पर अभी वो 10-15 लाख का खर्चा नहीं करना है।
तो फिर आप
आप भी
basically why you are going
स्टडी
for this all this, why you are going for all these studies? You do your cleaning validations for ROW with your only 10 ppm criteria, 0.1% criteria, which is giving you free values that your maximum dose is of 10 milligram. So 0.1% of the 10 milligram is this, and this shall not be present in another product. This liberty you are getting, then why you are going for this carryover limits and all these things?
अग्री
Because all
सर,
these
बट
things, if you
उसमें
want
क्या
to,
करना पड़ेगा, मुझे हर एक प्रोडक्ट में टेस्टिंग करना पड़ेगा ना सर।
वो
मुझे वर्स्ट केस ढूंढना है।
मैडम मैक्सिमम अलाउेबल कैरी, मैक्सिमम
हाँ, पर मैको